Thank you, James, and thank you, Dr. Ellis, for the invitation to speak today. And I want to first mention that our laboratories work with a number of different companies, and among them is Ion Torrent, and we've had some interactions with Ion Torrent. I will mention that technology today. But what I'd like to do is briefly discuss with you some next-gen sequencing applications, and then consider some other issues, such as preparing DNA libraries for sequencing, and then the steps that are involved in doing data analysis, and some of the considerations that those of us who are running next-gen in a CLIA laboratory have to face, keeping in mind that none of these platforms is FDA approved yet, so we have to develop these as laboratory-developed tests. Finally, we'll consider how we interpret variants and report them, and then we'll touch on some issues of specimen quality. So James has done a very nice introduction already over this, but basically we have two approaches that are being used, very broad-based approaches, for example, in the TCGA studies where we're combining whole genome, whole exome, whole transcriptome sequencing to really delve into the nature of specific groups of cancers. And basically all of these technologies emphasize discovery over cost and throughput. In contrast, in the CLIA laboratories, what we're seeing is, generally speaking, a more focused type of approach where we're looking at gene panels of, say, 20 to up to 400 genes at a time. These are generally limited to targets that we regard as actionable today in clinical trials. And we're concerned much more about cost and throughput than we are about discovery, keeping in mind that reimbursement right now is becoming a bigger and bigger problem. So Sanger sequencing is a way of assessing mutations and other alterations in DNA, sequencing across millions of pieces of DNA simultaneously and getting what is essentially an aggregate signal. In contrast, next-gen sequencing is what we call massively parallel sequencing, and each one of these reads comes from a single piece of DNA, of the original DNA. And so the number of reads that have a mutation, such as we see here, versus those that do not, gives you a specific allele ratio. It's a highly quantitative technique. And of course, the output is digital, which makes it a lot easier to work with. And now the kinds of boxes that are available for us to do this kind of sequencing are illustrated here. This is not the entire gamut, but these are two of the main boxes that are being used. On the side of doing whole genome, whole exome, and whole transcriptome, we're generally talking about the Illumina HiSeq. That's the one that's dominating the market right now. And when we talk about doing multi-gene panels, more in the CLIA laboratory setting, these smaller instruments are the ones that are coming to be used more often because they're cheaper and they're faster. On the Ion Torrent, the sequencing is actually performed on the surface of a semiconductor chip. On the MiSeq, the sequencing is performed on the surface of a slide or a so-called flow cell where individual strands of DNA are cloned out, and then the sequencing is, data is captured through a CCD camera. But regardless of how you do this, before you can sequence, you have to prepare a library. And this is true whether you're doing whole exome, where you want to look at all the coding regions, or whether you just want to do a panel of 20 genes. You have to be able to pull that target DNA of interest out of the genome, away from the rest of it, so this is all you end up sequencing. And there are two ways to do this, two approaches to preparing sequencing libraries. The first one is so-called hybridization capture. And in this instance, what we're doing is adding biotinylated probes that are made of RNA, and these will bind to the DNA regions of interest, and then those can be, after a hybridization period of about 12 hours, pulled out away from the rest of the genomic DNA by using magnetic beads that will bind to the biotin units. And so this hybridization followed by a capture is what allows us then to get to just those elements of the genome that we want to sequence, and these can then be adapted and put onto a sequencer. And again, it doesn't matter which of the platforms I've mentioned, this approach can be used for any of these platforms. The alternative is one that has more recently been introduced, and it's called the Amplicon-based approach. And here we're starting out with even less DNA, genomic DNA, and instead of using probes to hybridize, we're simply adding primers that flank each of the target regions that we want to sequence. We go through a short number of PCR cycles, maybe about 15 to 19 cycles, and then that becomes the substrate on which we're going to do the actual sequencing. So the final products that have been amplified away from the rest of the genome can be further modified with adapters and barcodes, and then they can be put on any one of the sequencing platforms that I've already mentioned. Okay, so here's a comparison then of the advantages and disadvantages of these two approaches. Hybrid capture requires anywhere from 500 to down to maybe 50 nanograms in the best hands. Amplicon-based, we can get away with even less DNA, 20 nanograms. How much is that? That's about 4 sections, unstained sections, of a 1 centimeter 20 gauge core biopsy. So it's not very much material. The hybrid capture requires overnight hybridization. The Amplicon, no, it's a little bit faster. The on-target reads, meaning you're getting what you want out of this process, is higher with the Amplicon-based approach. There is some noise coming through in the hybrid capture approach. Gene copy number can be assessed by both these approaches. That's actually not widely known, but we will be publishing on the Amplicon-based side shortly, and I'll show you some data that's not yet published. Now, how about fusion gene detection, translocation products? Well, you can pick those up by hybrid capture, not so much with the Amplicon-based approach. So this is a significant disadvantage for Amplicon-based approaches, and in our laboratory, we still use FISH as a backup for that. Now, when you look at what's being done across laboratories around the world these days, those labs that are doing whole exomes and large panels are generally using hybrid capture, whereas those labs that are doing smaller panels are generally gravitating towards the Amplicon-based approach. Okay, so we've prepared a library, we've gone through the sequencing. What's the next step? The next step is actually the much more daunting step. And it involves several different processes. So the first steps we've already discussed, sequence generation, but then we have to go into initial data processing, go through and identify variants and mutations in the DNA, and then interpret those and provide a report. Now, I've broken those steps out a little bit more carefully here, and you can see that the first step is what we call base calling. That is basically interpreting the actual signal from the machine and assigning it to be an A, T, C, or G. Following that, we have a whole series of basically random letters. The question is, what do they represent? So we have to align them against a normal genome and figure out this piece belongs to KRAS, this piece belongs to EGFR. Once that alignment has been done, we can then go through and carefully inspect to see if there are any nucleotide changes. These have to be kept in mind. It could be either mutations or polymorphisms. And at the same time, we're also going to look for insertions and deletions. And from here, then, once we've identified what we think are the changes, we can then do an in silico translation to see what would that look like at the protein level. In other words, what is the amino acid change that would result from that particular substitution? And once we've got that identified, we then have to go out and query the public databases. Has this variant ever been seen before? If it has been, is it a known polymorphism? Is it a known mutation? Is it a drug-sensitive mutation, and so forth. Along the way, we might also do a copy number estimation. That's a different process with different software. And finally, we're going to do a report generation. So it's quite a number of steps that are involved here, and actually many laboratories, I think, underestimate what's involved. Yes, you can buy some of these boxes for $50,000 to $150,000 and set them up and run in your laboratory, but you better be prepared to handle the back end, the analysis, because if you mess that up, you're not going to report the correct mutations in your report. Here's an example of copy number estimation that we've been performing on our 37 gene panel. There's 37 genes down here. Each dot represents the result of a single amplicon, and each gene that we're looking at, of course, is being covered by multiple amplicons, as illustrated by the different colors. This line represents diploid. So here we think that all of these genes have more or less 2 copies across the genome. However, in this instance of a breast carcinoma, you can see that the reads that we got from the HER2 gene are all way out of whack. They're much higher than you'd expect, and that correlates with the fact that this was a HER2 positive tumor by FISH. Similarly, we have a loss in retinoblastoma signals, so there's probably a deletion of one or both alleles there. And we have overrepresentation of a different kinase gene called DDR2 in this particular sample. Now, this kind of approach can be very helpful because we don't always run FISH on everything that comes into the laboratory, yet the sequencing can flag something as being abnormal. So here's a sample of breast carcinoma from a patient, and it was known to be HER2 negative, When we did the sequencing suggested that it was EGFR amplified. And I wasn't sure about that, so we went back and did the FISH, and sure enough, yeah, EGFR is very much amplified in this breast carcinoma. And that was a surprise to me. It turns out that there's a little bit of literature on this, but basically no treatment data to date. And I think it opens up an opportunity in this field. Now, for those laboratories that want to bring this kind of technology into their lab and run it as a clinical test, there are a series of recommendations that have been put out there. This is one paper from Nature Biotechnology. There are several others. The basic themes are that you have to assess the accuracy, the precision, the sensitivity, the specificity of the testing, and also take into consideration the reportable and reference ranges. So just to give you a quick idea of how we approach this in our lab, we assessed accuracy by using samples that have been previously genotyped by another platform, whether it was the Sequenom MassArray system or by standard Sanger sequencing. For precision, we looked at what we call repeatability, namely if we put the duplicate of a sample on the same run, do we get identical results? And then reproducibility, if we put the same sample on different chips on different days, do we get the same result? And can different technicians provide the same results from the same sample? And then we look at sensitivity by comparing allele ratios with another technology, or by doing a series of dilutions from a cell line where we know the original mutation and its allele ratio. And finally, specificity. If we see something that we haven't seen before, we go back and Sanger confirm it in order to make sure that it's real. And as it turns out, no surprise, with any one of these technologies, there are false positives that will come up, and these have to be understood and then subtracted out from future evaluations. This is an example of comparing allele ratios on the next-gen sequencing that we're doing in our laboratory versus a MassArray platform, a Sequenom system. And I have to say, when I saw these data originally, that was the first time that I became a firm believer that NGS will work. Because we can see that there's very, very good correlation. So that, for example, if I found it was about 75% KRAS mutation in a sample by this technology, I saw the same thing by the other technology. We also did things like comparing one sample run on a chip by itself versus one sample combined with others and barcoded. And was there any problem with that, interference between them? And the answer is no. We get basically identical results. And then we looked at true positives versus false positives and did a receiver-operator analysis. In our original data, we decided that a lower limit of detection that would be safe would be an 8% mutant allele. That would give us 100% sensitivity and 94% specificity. That is to say, there's still some false positives that are sneaking through, but those can be assessed by Sanger. We've since then, this was in the very early days, the software has improved dramatically, and we have now set the limit down to about 5%. Okay, so taking a look then at the output, this is the final output, the summation of all the alterations that have come out from just one 37 gene panel. And you can see that it's a fairly daunting list. Imagine what it would be like if we did this across, say, 400 genes or even 4,000 genes. Then the list is going to go right down through the basement. So when you look at this list, the first thing you can do is say, all right, we're not really interested in synonymous mutations. I mean, that basically is a nucleotide change, but it doesn't result in an amino acid change. So let's just not worry about those. But we then have these non-synonymous alterations, and the question is, what are these? And so we have to go out and ask public databases, are these polymorphisms or are they mutations? And it turns out that all the ones listed here in black are known polymorphisms. So then we're left with basically this short list of genes where we think that these mutations are real. They're not listed in the polymorphisms in the databases, and they're in genes that actually make some sense to us. So this happens to be from a head and neck squamous cell carcinoma. The tumor sample originally on the microscope had about 50% tumor nuclei and 50% normal cells. When we look at the alterations that we see here, there are two p53 alterations, each of them at about 25%, so we interpret these as being heterozygous. There's one stop codon in p16, and that's at 48%, so we interpret that to be homozygous, though the normal allele in those tumor cells has probably been lost. and these cells only have this mutated stop codon present. And finally, we have this mutation in KDR. So this is then the culmination, if you will, of all of the work put into the sample. We then put that out into a clinical report. Now, I asked my colleagues in oncology around our campus and elsewhere, I said, what do you want to see in a report? And they said, as little as possible. So I took that to heart, and I said, okay, if we're going to have a short report, let's just put down the gene, the mutation, and 1 or 2 or 3 sentences about what we think it means. P53, I think, is familiar to everybody. P16 is a common tumor suppressor gene often lost in head and neck cancers and many others. But what was interesting in this case was this, KDR. Now, KDR is the formal gene name for VEGF receptor 2, a well-known receptor tyrosine kinase. The problem was, when we looked at this mutation, it didn't exist in the literature. It's not listed in the cosmic database. There's nothing known about it. However, this is a patient who had failed all standards of care, was interested in getting on a clinical trial, and so we said to ourselves, all right, we think that this might be an activating mutation. We think it might be worth treating. And so we did. So this is the patient's tracheostomy site here with tumor growing out of it before treatment, and then we put him on a TKI with VEGF receptor 2 activity, and you see the nice response. Now, does this prove that this mutation is actually the main driver in this tumor and was the target for the drug? No, it's not direct proof. But it at least suggests that this might be the right target, and the next time we see it and we treat it again and we get another good response, then we can start to build up a database that gives us more confidence that we're doing the right thing. Now, a couple of things we have to keep in mind is that all of the sequencing that I'm discussing and all of the data that comes out of it is directly dependent on the quality of what goes into it. Garbage in, garbage out. So if we have a sample that's sent to us like this, I'm one of those rare individuals who will look at a sample like this and say, great. I mean, most people look at it and say, oh no, that's terrible, that's metastatic melanoma in the lung. But I'm saying, no, 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 this is good because we've got a perfect nodule here of tumor, very rich in material that we can puncture right down into the block and get basically 90% tumor cells out of that. So I'm excited. And many samples we can do that. If we have this much material present in a sample, we will go ahead and punch it, and as illustrated right here, and that just gives us enough material out of the paraffin block to do the sequencing. But most of the time, we are a little bit more careful, and we will actually outline regions of tumor on the slide, and then the technicians will scrape those areas selectively, and that will give us the most enriched material. This is time-consuming, it's labor-intensive, it's not terribly well reimbursed, but it's necessary, because if we don't do this, we're not going to have as much tumor as we'd like. And here's an example of one such type of situation, metastatic colorectal carcinoma in the mesentery. These are the only tumor cells that are present, these little clumps of purple. I can have the technician focus just on this region of the slide, but honestly, I'm going to end up with only 10 to 20% tumor cells. This is the lower limit. I'm not going to go anything lower than this because I'm afraid I will miss a mutation and not do the patient a service. And so we're very careful and very selective about what goes into the laboratory. The most disappointing samples are those that come from transbronchial biopsies. And here's an example where this fragment, no tumor, and this one down here, teeny focus of tumor, but really that is just not enough. So please, we have to get another sample from this patient because this is not enough to do it. Now, two other things I'm going to just mention in closing. One is that we have to be aware of the effect of decalcification. So if a patient has a bone biopsy or bone marrow biopsy, and that is put into acid in order to soften the bone and allow a section to be cut, that will destroy the DNA, just irretrievably. There's just nothing that can be done for it. The alternative is to use an EDTA-based solution, although that's not quite as effective as a decalcification procedure. So if you're working with bone biopsies, for example, of metastatic breast carcinoma in the pelvis, you want to talk with the with the pathologist and the radiologist and say, look, just try to get some of the soft material without decalcification into the block so that we can use that for sequencing. Because if you decal the whole thing heavily, we'll never get anything out of it. The other thing to keep in mind, older material. So if you have paraffin blocks that have been around for 4, 5, 8, 10 years, the longer they've been around, the more we run into this problem of deamination. This is a spontaneous problem where cytosine amino groups are lost and convert into uracils, and this basically mimics a mutation. So to summarize what we've discussed, NGS has major advantages over traditional assays. Very good sensitivity, requires very little DNA, less DNA than a typical KRAS test. And we can detect copy number alterations and, in some cases, translocations. However, we have to keep in mind that there is no such thing as a perfect test, that sample quality is important, including the tumor content and how it was fixed. And we also have to keep in mind assay limitations such as false positives and the challenge of finding some of the longer types of insertions and deletions, which at the software level is still a big challenge for us. This is where I would say we have a weakness that has to be addressed. So thank you for your attention.